Okay.
I don't deal with that.
Jenny, let me ask you one other question. So, have you ever noticed a difference in variation between lab to lab? Like you send the same soil results, same soil to the same different labs, and they're all over the fucking map.
Yeah. Yes.
How do you compensate for that?
Should ask somebody outside the laboratory sector.
Now don't get yourself into trouble, Jenny. Okay.
You need to look at secondary witnesses. You need to, because the plants tell you this, the soil tells you. If you're sending water or soil or sap or tissue out just because somebody told you, then you're just going to get the results and you're probably not going to do anything and put it in a file. And I understand that. But if you're doing it to learn, I would send multiple tests. I would do multiple sap, tissue, soil, Haney, water, very important. I would look at them together because they work better together anyway. And if there's always like one crazy outlier, then I would call and be like, how are you getting this? What does this mean? How are you getting here? And just use it as a learning opportunity because you could be looking at it differently. They could be humics, testing of humics. Are they putting in ash? Are they not putting in ash? Humics, 16%, probably not a normal humic percent at all. Humic at 9%, yeah, that's probably doable, absolutely. But there is no criteria for, are you going to ash it and subtract out the ash or ash it and keep the ash in? I don't know the whole analyses, but I do know owners of humics, and I know the director of the Humic Fulvic California Society, however you mix those words up. And the testing is different, and understanding humic or ash or no ash. If you are looking at anything with a spore in it and you get these astronomical counts